So as we come back, we're now going to transition our discussion from burdens and impacts in daily life of CMT and inherited neuropathies, and we're going to start shifting to the discussion of current treatments. But before we do that, we also wanted to set the stage a little bit with a pipeline overview of therapies in development for CMT and inherited neuropathies. So to lead that presentation today, we have Dr. Florian Thomas, who himself for 20 years has practiced interprofessional care to help people with CMT. And in fact, in 1998, he founded, with patient advocates, a CMT support group. So among other things, he's a professor of neurology and founding department chair at the Hackensack Meridian School of Medicine and Hackensack University Medical Center. So please join me in welcoming Dr. Thomas.
Thank you. Let me start by thanking the FDA for having this event, which I think is very useful. I also want to thank Alison Moore and the entire H&F staff for such a great organization. You may notice that some of the researchers and pharmaceutical companies that are seeking to improve how we can care for people with CMT are here today. And I think that's a good illustration of the two-way street that is required for successful outcome. How we test drugs and how we choose outcome measures has evolved greatly over the last 20 years. FDA doesn't want to look at drugs anymore that don't have quality of life outcomes and have patient-reported outcomes. And that's a very important development. And I think it recognizes the degree to which patients with the condition bring a lot to the table. And of course, this meeting is an expression of that. So I wanted to talk about a couple of developments in the CMT world that reflect our better understanding of the disease, of the genes that are involved with it, as well as a development of technology. Many of the drugs that we'll be discussing would not have been possible 5-10 years ago. So I think it's a good thing that this talk will very soon be outdated because new things are happening in the field every day. So I want to start with in declaring my conflict of interest. I'm an investigator both with Acceleron and with Pharnext, and I also have an early involvement with the HDAC compounds that we'll be discussing. A083 is a locally acting muscle therapeutic which is in a phase 2 study of people with CMT1 and CMTX. As shown on this slide, it serves to upregulate muscle protein production by blocking an inhibitor or various inhibitors of muscle protein production. And the goal is to verify if this mechanism can serve in patients to strengthen their tibialis anterior muscles. It's already been tested in healthy volunteers and was shown to increase muscle volume. And it's also been tested in people with a very focal muscle disease where it is similarly applied to the tibialis anterior muscle. These are the typical outcome measures that we are testing in patients. So various measures of muscle function as well as muscle volume. Muscle volume we are examining by MRI. We are testing both strength as well as functional measures such as 6-minute walk test, 10-minute walk, run, and so forth. The preliminary data that we described in July include that indeed in people with CMT we were able to show an increase in muscle volume as well as specifically contractile muscle volume and a decrease in the fat volume of muscle. So this suggests that as the muscle that we inject, the tibialis anterior, which of course is paramount in its impact on ability of people to walk, the contractile muscle function of the tibialis anterior muscle improves. So that supports continued investigation of this compound, and so we started phase 2 of part 2 of the study, whereas part 1 was a dose-finding study. It allowed us to select a particular dose for phase 2, and that we hope to complete in late 2019. And we're actively enrolling patients for that. The PXT3003 agent is an interesting compound which is the result of a lot of what we call in vitro testing, where we examined for patients who have CMT1A due to a duplication of the gene, what we can do to reduce the amount of the PMP22 protein that is made. And we found that there are 3 agents that we can use together to achieve that goal. And those 3 agents work on 3 different receptors, and they are drugs that are in common use for many decades, including baclofen, which we've used as a muscle relaxant, naltrexone, which is an opioid antagonist, and sorbitol. And we use these 3 drugs at a fraction of standard doses. So whereas you might treat a person who has spasticity from multiple sclerosis with 20 milligrams 3 times a day, in this trial we use it at a couple of milligrams twice a day as a cocktail that a person takes twice a day. And the goal is by reducing the amount of PMP22 protein towards more normal levels, one can halt or revert the disease process. So the study design of an earlier study that was published in 2014 enrolled 80 patients. It was placebo-controlled. It was given twice daily. We measured fairly standard outcomes that are often compounded measures of symptoms and signs, motor symptoms, sensory symptoms, and signs. And we also looked at how the electrical function of nerves was impacted. So if you look at the green line, is there a pointer here? How do I activate that? Okay. If you look at the green line, which is one of these compound measures of strength and sensory symptoms and signs, you see that there was a statistically significant improvement, whereas at the higher dose, whereas at the lower dose there was not, and with placebo there was deterioration. So that phase 2 result was very encouraging, and it allowed us to embark on a pivotal, where pivotal means that a study is designed to be submitted to FDA study. So we designed this with many more patients and similar outcome measures. The study is being done in Europe and in North America. And we are waiting with bated breath for results which are to be released late next month. So this compound was tried in patients aged 16 to 65. This study of Flex 787, a co-activator of 2 compounds, is an illustration of how our better understanding from talking to patients as to symptom relevance has impacted treatment approaches. As I was discussing earlier today with some patients and colleagues, in my experience, the pain and cramps of patients are something that patients rarely volunteer as complaints. But when you ask, and I think the numbers when we took the survey, the polls today reflected that, there are quite a few patients for whom this is a major disruptor of daily life, both during waking hours and during nighttime. And of course, when a person's sleep is disrupted, that has a significant impact on the sense of well-being the following day. So it was important to see if we can alleviate that symptom in patients. That's what the study sought to achieve. Unfortunately, it had to be terminated because of the taste of the agent. Wasn't tolerated. For about over 20 years, we've tried to apply various methods to correct diseases by inserting better forms of a gene into cells. And there are long-standing approaches that use different types of viruses to achieve that. By inserting a normal form of a gene into a virus and removing from the virus any kind of sequences that could be harmful. So this has been done for a long time, including in CMT, but this approach is now reaching maturity, as this slide shows, where the injection of a viral vector with a normalized gene into the spinal canal can target particular types of CMT, particular cells that harbor or that express the gene, the mutation of which leads to the disease. So this approach is underway for giant axonal neuropathy, CMT4J, GARS mutation, CMT6, and X-linked forms of CMT. And as you can see, the arrows point towards phase 1 for giant axonal neuropathy and are in much earlier stages for the other 4 types. The histone deacetylase inhibitors are an interesting group of agents which seem to be able to play a role in a potentially wide range of forms of CMT to allow for better maintenance of the length of the axon. So as shown on the lower half of the slide, axons, the neurons are very specialized in the peripheral nerves. The cell bodies sit within or just next to the spinal cord, and then you have this endless axon, which can be, depending on the size of the person, up to a yard long. And that is an unusual cell type. Most cells in the body are fairly round, and that means that the challenges for maintenance of that cell are very different. In axons, you have to carry appropriate organelles that maintain energy supply in your toes all the way from the spinal cord. So that's very challenging, and under ideal circumstances, this works well. But if the axon isn't healthy, for whatever reason, and of course the reasons are different in different subtypes of CMT, there are problems at the end of, in particular at the end of the nerve. And that's why many forms of CMT are length dependent. That's why people have weak toes before they have weak ankles before they have weak knees. So with histone deacetylase inhibitors, the goal is to correct the maintenance of the very long axon through what we call intermediate filaments. There are a bunch of agents in development that seek to promote normalization of axonal transport, which, for instance, includes mitochondria, which are the power supplies of our cells. So as you can see in the lower slide, in the lower part of the slide, the goal is to transport these mitochondria to the end of the nerve so that the end of the nerve is just as healthy and just as much energy that it needs as the cell body in the spinal cord or next to the spinal cord. So initially, this approach is being applied in animal models to the equivalent of CMT 2A CMT 2D and CMT 2F, but other conditions that are also axonal may follow suit over time. So there are two companies that work on that. So this is further illustrated here, where the microtubule-based axonal transport we think is impaired in these conditions. So by enhancing the health of microtubules, one can maintain the length of the axon. As discussed already in a couple of talks, in the single most common type of CMT, CMT1A, due to duplication of the PMP22 gene, there's not just too much gene, there's also too much PMP22 protein. And the dysregulation is felt to be the cause of the neuropathy. So are there ways to reduce the increased amount of protein? Well, we've discussed one way already. PXT 3003 seeks to accomplish that with what we call a polydrug of baclofen, naltrexone, and sorbitol. But there are other ways to think about correcting the overproduction. So we found an agent called P2X7, which is a binding partner to PMP22. And PMP22 induces an increased expression of P2X7. And together, those two proteins forming a complex may be responsible for Schwann cell death, and Schwann cell death damages the axon, and that's why one gets a length-dependent neuropathy, CMT1A. So there is an antagonist, a compound that counteracts P2X7, so-called MT002, and that may serve to downregulate the PMP22 production in itself, thereby restoring levels to normal and hopefully improving peripheral nerve function. Another approach that is very exciting because it is already on FDA approved and in clinical use for another very rare neuromuscular condition, spinal muscular atrophy, seeks to correct genes expression through RNA molecules which can change the amount of RNA that is converted into protein. So an antisense oligonucleotide, ASO, to PMP22 would, at a specific dose, reduce the amount of PMP22 RNA and therefore PMP22 protein, And this concept has been validated in rodent models of CMT injected into the skin. Obviously, patients would rather take medications by mouth, but there are lots of medications that would be broken down if taken by mouth. So I already talked about the viral vector that's injected into the spinal canal. This agent here is injected into the skin of the rodent models. So stay posted on that, but it's certainly another very exciting mechanism. There are many more drugs in development in different stages. One of them is a product by Inflectus, which seeks to improve cells' ability to deal with misfolded proteins. So protein misfolding is another mechanism where a mutation in a gene changes how that protein is handled, and proteins that carry such mutations can accumulate in cells and damage the cells. So that study in CMT1 is expected to begin in 2020. Another company, Cydan, is committed to Charcot-Marie-Tooth disease as well. I wanted to not just leave you with a discussion of medical or pharmacological approaches to CMT. There are lots of other open questions to CMT and how one can improve outcomes in patients. Several of the audience members discussed how they fared with surgery. There was a meeting in 2016, published in 2017, of foot and ankle surgeons and CMT specialists to figure out what is the best surgery, what is the best timing of surgery, should one wait longer or less time, and how do we measure whether surgery is successful. There are no good parameters for any of these three criteria. So there's an active study underway, and I think one of the next speakers, Dr. Pfeffer, is going to discuss his long experience with foot and ankle surgery. Another approach is to teach people to do more with less. So how can we improve balance even when the peripheral nerves are no longer in a position to tell the brain where your foot is? Can one train people to use the reduced cues as to whether the ground is even or uneven and respond appropriately? And there's at least a pilot study, pilot because it was only 14 patients, that was published 2 years ago, that seems to show that that is indeed possible. And of course, falls, as was so amply discussed by people today, have multiple bad consequences. They can lead to fractures, they can lead to embarrassment, and some people also discussed the difficulty feeling the accelerator and brake pedal. Can one improve on that? So studies such as this have major impact on functioning and quality of life of people with CMT. So that's what I want to discuss today. Thank you.